This is your professor again, and this is your last lecture. And I promise it will not be 37 minutes. That one was way too long. Um, okay, so it's animal development, and I've got like 10 slides. I swear it's going to be less than 10 minutes. Um, so here's some questions that I want you to be able to answer. What are differences in the stages of development? How do different stages differ in their physical characteristics? And you'll know exactly what I'm trying to get you to think about as I go through this presentation. What are mechanisms? I probably should say mechanisms associated with fertilization. We'll talk about that, and transitions from zygote to organism. And we'll cover that a little bit. I mean, it's mostly descriptive. Okay, so continuing. Here are the things. I like this. I like this line here, and I like that here. Fertilization is a process that creates a thing, right? A zygote. Cleavage is a process that creates a thing that is commonly known as a blastula, but as you will see, they're not always called blastulas. And then there's gastrulation, a process that produces something called a gastrula, and then organogenesis is something that creates embryo, and then embryo hatches out or born, produces an offspring. Okay, so let us continue. Gametes, yeah, you should probably know a little bit about gametes, right? Sperm and egg, small, built for travel, right? That's sperm. Millions of them every time an ejaculation event occurs, whereas there's only one egg, and all your eggs that, if you're a mammal anyways, all females, either you have more eggs when you're an embryo than you are when you're alive as a human, living, crawling around baby. And then the numbers diminish as you get older until there are none left. They're large, contain nutrients and things called cytoplasmic determinants, which include messenger RNA, and these cytoplasmic determinants are going to affect the fate of cells that are produced during development. One of the big questions in development, of course, is like, how do you go from a single cell that is identical to all subsequent cells in its genetic makeup, but all subsequent cells are not doing the same thing, right? And it has to do with genes being turned on or turned off. And some of that is due to what's inside the cell already at the beginning, cytoplasmic determinants. Egg contains organelles. In mammals, there's also the known as pellucida extracellular matrix, where sperm binds to receptors, and then a collection of cells that support the gamete, the egg. One of the big questions that happens during fertilization is why only one sperm cell makes it into the egg. And this is a lot more information on this slide than I really want you to know. I'm gonna first say that there are two mechanisms that are thought to be involved or known to be involved in affecting the ability of sperm to penetrate the egg after the first one has made contact with it. There's something called fast block of polyspermy, which has to do with a polarity change in the cell membrane, and the possible mechanism here is that the sperm requires a certain charge on the cell egg membrane for the sperm to actually be attracted to the membrane. But the only thing I'm interested in having you know is that there's this fast block which happens immediately after the first sperm molecule cell binds to the egg. And then there's something called slow block of polyspermy, and this is involving little granules, vesicles just underneath the cell membrane. And these granules contain enzymes that, once activated by calcium, and you can see here calcium activity increasing after the sperm penetrates the egg, calcium triggers the release of chemicals from these vesicles, and the vesicles once having the chemicals released, will do all sorts of things that make it impossible for any other sperm to bind, including the removal of sperm receptors on the surface of the egg. And I'm not going to test you on this in any depth. I'm just going to ask you, like, what are the two mechanisms that are known to be associated with controlling the penetration of sperm into an egg cell? One is involving changes in the polarity or the membrane potential, and the other is involving the release of enzymes by these two things here. Fertilization. Fertilization, and this is sea urchin, so can't really say that this is true for everything necessarily, but sea urchins, which have been studied because of their the ease with which the process can be observed under microscopes. Calcium triggers activation of the egg, increases protein synthesis and cellular respiration in the egg, and subsequently there's fusion of the sperm nuclei with the egg nuclei, and you then get DNA replication and the first cell division. These are the events in fertilization of an egg by sperm in one particular animal example. I might ask you to tell me what the events are in order on exam. Probably not. Okay, so we talked earlier about these events here, right? Fertilization gives rise to zygote, cleavage gives rise to blastula, etc. So now we're talking about cleavage. And I want to begin by saying I'm not interested in this slide, so we're moving on. I am interested in this slide, but only in the following sense. So cleavage patterns, and I say frogs, but it's actually there are cleavage patterns. There are other patterns that I want to mention here. The blastula, when we think about a blastula, the classic developmental stage in animals, that is really a frog thing. There are other types of blastula-like things, blastocysts in mammals and blastodiscs in birds, which I'm going to talk about in just a moment. They are about the same stage. And what does this stage mean? Well, classically, when you think about blastula, you go from a single cell to a multi-celled thing. Could be hundreds of cells. It's all the object is still the same size. So all you've had is cell division. And one of the consequences of this, if you go back to cytoplasmic determinants, is you start partitioning out some of these cytoplasmic determinants into different cells. And so the cell fate is already being determined as early as the cleavage, during cleavage, because of the movement of these chemicals differently into different cells. Of interest potentially is that up to 8 cells in humans, and I think probably in other groups of animals too, cells are totipotent, meaning that you could break those 8 cells up and you can generate a new offspring. And that's one of the reasons why when you hear about octuplets, it's because of that. Okay, moving on. This is a blastocyst. This is a blastodisc. And in the case of a blastodisc of a mammal, this is the actual blue here, the inner cell mass, is what develops into the embryo. And the other parts are going to form the placenta. So here's an illustration of what's happening. Egg is fertilized in the oviduct and then makes its way. Cell division is occurring. We're going through cleavage and eventually getting to blastocyst that will embed into the uterine wall, and part of this becomes placenta, and part of this becomes the embryo. And this is a blastodisc. Blastodisc in a bird is interesting only in the sense of, like, if you've ever seen an egg, a chicken egg that actually has a little white halo thing on the yolk, that's because that's actually an embryo, a blastodisc. and that egg is fertilized. Gastrulation is the next process, and then after gastrulation is neurulation, and then we get to organogenesis. And I'm not really going to spend time talking about all these things, because this is stuff you can just draw off. What I want to have you remember is the following. There's two things to remember here. One is that all this stuff that's happening in gastrulation and neurulation and organogenesis involves specialization of functions of cells. So we get cell determination, differentiation happening in these stages of development. So again, you go back to the original cell, had all the same genetic material that all these subsequent cells have, but all these subsequent cells are doing different things. And how does that happen? How does some cell become a muscle cell and not a nerve cell, or vice versa? That's the second part of this story. And the second part of the story is that where a cell is in an embryo will influence what it becomes. And that word that you want to remember here is induction. Dorsal lip is part of this embryo. It induces notochord formation. So there are chemicals that are being released by cells in the dorsal lip that are going to affect what the cells neighboring to it will become. These cells then subsequently induce something else to happen, and it's very common for cell fate to be determined by a process called induction, which is communication between neighboring cells that will affect what genes are turned off or turned on, to then subsequently give rise to different kinds of proteins and structures within those cells and determine what they become. So as I said, there are really only a couple things I want you to know here about gastrulation, neurulation, and organogenesis. First is that we start seeing specialization of cells, and the second thing is that that specialization is a product of, at the very least, there are other things involved here, but the one that I want you to remember is the process of induction, inducing cells to become something because of where they are in the embryo. And I think that's all I want to say. That's it.